now we're going to change session. We're going into roundtable mode. And so that will be the moment where, if you have questions, prepare them. If you can write them in the chat, the team, I guess the facilitators from the BioSummit will, check them, will remind them, and so I'll be able to ask those questions to the speakers. So I'm going to start actually with a few questions, more general questions coming from myself to kickstart this roundtable. The first one would be, you've been working distributedly through the entire length of this program for the last 6 months. And so I guess a lot of people want to better understand what it takes to make this happen. Like, how have you been managing your work? What methodology would you recommend others to apply to be successful in this kind of approach? Anyone can start.
I think the first thing is that JOGL gave us this framework for, you basically said, here, you can meet twice a week. These are the slots. You gave us the Zoom channel. We knew that everybody else on other projects was meeting. So there was almost sort of a peer pressure to continue meeting. And in a weird way, I think just setting up that framework where there was an expectation that we would talk every week, no matter what, was one of the key things.
Would you like to complement?
I guess I could just add, without naming names, but I was involved in, we've been trying to get collaborative science to work across many labs for a while now, and not very successfully, I would say, in most cases. And I know I was involved maybe a year ago in one that was led by someone who had a community lab, and maybe 4 other labs were involved. And the experiment was a scientific experiment in itself, but also the bigger experiment was, can we make collaborative work successfully across many labs with different kinds of equipments and different people at different levels and doing this biohacking stuff for different reasons. And we were not successful. And it was a failed experiment. And no fault, I'm not faulting anyone involved, but what I see is different with JOGL and why it's been successful. I'll try to be brief. I know a lot of people want to talk. But I feel like some of the things are that, like Ellen said, we have a neutral platform where we can just jump on and we discuss twice a week, whoever can make it. And then the funding was key. Really, it's not a lot of, it's micro grants, but for a lot of us, for my community lab, it meant everything. It meant doing the project or not doing the project. And so that component, I think, is new. It's not really something I've seen to foster collaboration. Anyway, and it's just a great group of people that are, the Slack channel is super active, and I don't know, just is a great place to bring like-minded people together in a neutral place.
I guess I could also add that the special thing was also that we were all in the middle of this pandemic. We were all in lockdown. There were all these preprints coming out and so many things to read and discuss. I felt like I was a graduate student again, it was so exciting. And we ended up being so busy with like 5 different Zoom things every week, and finally getting funding so we could get these molecular biology tools and do the R&D. It was incredible. We're really very grateful for this opportunity that came from such a tragic thing, but maybe it'll help us be prepared for the next pandemics too.
One other thing I think was very useful was actually Slack. Slack, in a few different things I've been involved in, has proved to be really, really brilliant for communicating.
And it was so cool. It was almost like everyone was trying to be the first one to post anything new that we saw to the group. You know, it turned into a very sort of friendly competition as to who could provide the most value for everybody else in the rest of the group in a weird way.
I also liked, I like that we have, well, weekly or semi-weekly now, but meetings as a JOGL group as a whole. I think that's also motivational. I mean, because then we're asked, like, what did you come up with new in the last week or in the last 2 weeks? And then, you know, it's the same idea. It's like, no, you want to have something to put in that slot on the doc. I mean, because you know, you want to tell everybody what you've done. And
yeah, it was the best kind of friendly competition on a neutral platform.
And so, what would you have wished to, or you know, looking at how things were beyond the things that worked, like what would you say either didn't work very well or actually were missing? Like, do you see something that will make this kind of collaboration even easier?
It was weird because we had sort of both the JOGL platform itself and the Slack channel. And right now there's sort of a link between them, but it's not fully integrated, and I don't know anything about Slack and, you know, what you can do with it or what you can't, or how restricted it is or how open it is as a platform. But it's much easier for people to use something they're already using in their business or in their personal life, and Slack is used by a lot of people and they're familiar with it. I don't know how you could integrate it better into JOGL, because right now the JOGL pages are mainly for putting kind of what our progress is and sort of an entry point for people who want to join projects. So that really, I think that's my biggest thing. You know, people will say, well, I joined JOGL, but how do I get on the Slack channel? So something very simple and mechanical, really.
Yeah, I guess just as an aside, I mean, our JOGL interactions, you know, and how JOGL has been successful is actually the subject of an NSF grant at the moment. So, I mean, I think that's pretty cool to mention for Thomas and the JOGL organizers as well.
Yeah,
we never would have found each other in a million years without JOGL. That's the way I feel.
Yeah, and I think, you know, with the pandemic just coming on stream, it was pretty amazing how people around the world converged on JOGL. And, you know, for those that don't know, if you go and explore the different types of projects, there's just so much. There are people building respirators, you know, PPE, all sorts of things. So it's not just what you've heard about here in the talks today. It's so much more. And people sign on. And it can be quite overwhelming. I think that's one of the big challenges for new people, but it's been really good to see how JOGL's team have actually been able to adapt and recognize that there are challenges and then create some kinds of solutions that, you know, onboarding of new people, things like that. So, you know, just like David says, you know, this is an airplane that's being built while it's being flown. So
yeah, I mean, I reiterate that super flexibility
and under the pressure of a pandemic.
I can add that, yeah, like we tried to put a lot of work into onboarding, even like internally in our group, and made documents and stuff, but in the end we're still, most of the team is still the core team that was there in the beginning somehow.
Yeah. So
I think we all need to learn how to better be able to get more people in and actually get how to build it up.
Yeah, from my side, it's we have seen when this German hackathon was onboarded to Slack, the first week had 40,000 users. But there were 100 moderators to make sure that the community is not collapsing, not fighting with each other. So that's the greatest aspect. Like, JOGL had that from the beginning, that how to have the code of conduct and how to take care of the community issues and how to moderate that. Like if someone is like if they don't know how to go, where to go, and as Ellen said, that it was initially a little bit difficult for people to be onboarded from the JOGL platform to the Slack, and then it was later done perfectly. And also the good aspect of that is they were every week in the community, the meeting, the statistics were shown to the community, how good the community is growing and which are the topics which are more trendier within that Slack, so that people would just also know something which they didn't know initially. And so that's the whole connecting the dots is a very important aspect in building virtual and real physical communities. And that's what JOGL was doing very well.
Well, I'm honored by all those feedbacks. The question now is, where do you think, where do you see those, where your project, but more generally, these kind of collaborations go in the future? And do what do you think is the impact of the pandemic on open science and community science?
Well, I think it's just a great example, a great model to follow for future research. I've had that question asked in a few different ways before from different people, like, where do you think research will be different now for you? Do you think, JOGL will continue after the pandemic? And I mean, yes, and I hope so, but it's a great model for, if we can sort this out. I mean, I hope I stay in contact with all of you guys, after we've passed this huge, horrible hurdle and we're better. But, I mean, everybody on that I have interacted with is amazing as a scientist, and I feel very fortunate to be connected to everybody on JOGL, and I hope that we will be doing many other projects together. And this is a great way to do it. Like preprints were mentioned by Ellen earlier, and I mean preprints, there's so many, and we read them all, like, right? And we tried most of them, and and it was a little bit, once we realized a lot of them weren't reproducible, unfortunately, it was a little bit like, let's go through and see which of these look feasible. We would do that and then try, a few of those, but is sorting through the data kind of as quickly as possible. And it's like a Google search, but it's like a preprint search, like we were doing, kind of thing.
I mean, it certainly helped to have a lot of brains. Like if you hit a snag, you could throw it out to the group, and somebody might have some insight on it from their previous work. I mean, it's the way that collaborative research should be done, but often isn't, due to all sorts of boundaries around the way people are, get their jobs and are promoted and start companies, and the proprietary nature of things. So that was very refreshing. I think a lot of people hope that science will become more collaborative in general after the collaboration we've seen, not only in this group, but worldwide around COVID-19. But as we all know, there are forces working in the opposite direction. I mean, I think the big elephant in the room is climate change going to be tackled like this too?
Yeah.
And a lot of us wonder. But one thing I just want to throw in is that we are also getting boots on the ground reports. I mean, we would always say, well, what's happening in Paris, what's happening in the Netherlands, what's happening in XYZ place, like, are people wearing masks? Are people not wearing masks? Are stores closed? Are they open? Are people frightened? And to get a firsthand account from the people in cities all over the world in real time, I think is another thing that we don't talk about very much, but to me, it was really, really informative.
Another thing that I think that I see coming out of this is more reproducible explanations of how people actually go about doing their work in the various labs. And even things like protocols.io, the way the X Prize has asked us all to put all of our protocols up on there, means it looks more reproducible for other people to check it out. I think that's something that might come out of this as well.
I think also another thing is that, you know, beyond us as kind of like trained molecular biologists, I think the pandemic has really kind of, you know, like everybody knows about PCR now. That's kind of weird, right? Like everybody, you know, and just this kind of overall global education of, you know, virology and molecular biology. And, you know, I think has the potential to contribute to different kinds of science that we might engage in the future. That's been really interesting for me to observe, just like amongst my community.
Slowly but surely. PBS NewsHour did say Regeneron's antibody was polyclonal at first.
Yeah, so but this is exactly very true for us, because Sri Lanka is known as an island or country for tea and tourism. I think this is the first time, probably even for SLINTEC, the institutions, is the first time some form of a biotechnology solution has been locally made, locally at least prototyped. And that was possible only through JOGL platform. And I think the barrier, I mean, there's so much a program like AGiR, but the barrier to these programs are very, very high. Like, you know, we discussed this even in the Asia regional meeting, like $5,000 is a big money for us, for our community. So barrier to such knowledge and infrastructure to the network through JOGL was very easy. And then we were able to tell a lot of community students that, you know, they could learn easily. And I hope that could be also that we have more such success stories like Sri Lanka in other parts of the world, that, you know, to enable local people to solve their local challenges. And that should be the great model for JOGL in the coming years.
Yeah, exactly. I agree with Ara also very much that having been involved in all of this really gives more hope for open participatory research to solve problems in the future. I mean, certainly all the things that we've put out could be manufactured anywhere. Someone could maybe even make money off of it and sell it, and we'd be perfectly happy. And the idea that you don't need to have IP necessarily to actually make a profit when there's a market for the thing is something we always talk about at Hackuarium. And we want to see more of this sort of thing happening. And I think it's great that Ellen brought up the point about climate change too, because we have so many problems on our plate, and we can't let politics keep us away from them. And when this kind of global community gets together to do something, well, who knows where we can go. I really it brought a lot of hope to me just to be participating with this group.
Yeah, and I just wanted to add, so I dig into the IP or patents for BST wild type. And it's just crazy because, like, for the NEB patent, for example, you have 400 different sequences in the patent. So it's not just like protecting this one product. It's like also, it's kind of a warfare, yeah, kind of a weapon in an economical sense for, yeah, getting competitors out of the game. And it's not okay, because most of the biotechnological knowledge came from nature, and nature is like our common thing, you know. It doesn't belong to anybody, and it's not okay that you just don't patent this one sequence, you patent so many others, you know, it's, yeah, freaked me out.
Does anyone have questions for amazing speakers? Don't hesitate to put the questions in the chat.
Actually, Kevin is raising the hand. Maybe like we can allow him to speak. Kevin, can you?
Sure.
Oh, yeah, okay. I
think so. Hi, everyone. Good to see you all. I joined a bit late, so I'm sorry I missed the earlier thing, but this is wonderful to hear. I wanted to ask about, I guess, looking at these open COVID projects, like in particular, but this applies to other projects in general. But bringing these products into, like, I guess, competing with industrial work or with academic research, or like actually getting things to market, kind of thinking about how your work benchmarks against existing work that's done out there. Like, I'm wondering, you know, people will talk all the time about like the latest company doing the latest best drug or the best COVID test. Where are you guys on that kind of map?
I would say right away, we're not trying to compete with companies. I mean, most of us want to help locally where we are, or help communities that don't have access to those fancy drugs. So, I mean, I can tell you right now in New York City, yeah, you can get a free COVID test, but you won't get the results back for a while. And you have to make an appointment. And you won't get an appointment sometimes for a few days. So there, you know, this availability of testing is just not there. And so, I mean, this brings up a weird point, and that's some of these techniques like isothermal amplification are really easy to do from a technical perspective. So the regulations have changed a lot just since January. We went from this emergency use authorization where you needed a lot of paperwork, and I'm talking in the United States. I don't know what the situation is in other countries, how much freedom people have. But now it's got down to the point where if you call it surveillance and you say that someone has a potentially, what is it, a likely clinically relevant result, and you don't call it a diagnostic, the door is almost open for anyone to test anyone, and certainly for people to test themselves. So in a weird way, I see some of these open source protocols going almost in that direction. I guess that's controversial, but it's just what I see.
So I just saw Carolyn's question.
You're muted, Rachel.
So I was just going to go in to answer Carolyn's question about governments being willing to use such solutions like this. And Sri Lanka, the country of Sri Lanka, is really quite sold on this whole idea already. And I think actually it's not necessarily governments that will always be the ones working on this sort of thing, but if, say, we get to the X Prize finals, then it'll be this sort of multinational sort of thing, this contest that will spur on more development of our methods and help them spread. And yeah, depending on how well we do with this blinded proficiency test that we've been waiting for with bated breath for a couple of weeks already, I think it is possible, not just that they'll look at us and JOGL in our open science stuff, but that they'll see, aha, they did really well on this blinded test, and see how things work in the finals. It's really like over 5 weeks, over 500 tests per week, something like that for all the kids. So it's really a sort of validation that someone from an open public lab could never organize. Doing sort of clinical trials is super expensive.
Well, the access to the French public health system that Thomas is providing can actually potentially provide us with some sort of clinical validation, which obviously from an ethical point of view, we would have to have some clinical validation data to allow people to be able to judge whether or not what we're doing in terms of testing is valid or not, regardless if they were, you know, asking for help with it or just taking the protocol and doing it themselves. I'm not comfortable with putting something out there and just saying, let the user beware. I think it has to be put out there with some data, which is why, you know, we haven't really made that much noise about our protocol. And the Sri Lanka, it's being used in Sri Lanka with validation with clinical samples. That's the big difference.
Yeah.
And to add something a little bit also, like what Rachel mentioned, inside Sri Lanka, you know, so it's a colonial country, so they always look for, it's an island, they always look for opportunities, something they can export. So when I presented this, every time the question was, okay, can we sell this outside of Sri Lanka? You know, can we like make money out of it, even for government, because it's an island nation with, you know, with not much, you know, resources. And so I said that no, actually we cannot, first of all, because it's an open source, and then we have our exclusivity to our country. And then they initially, even for government, even for like traditional institution, they initially, they never heard of such thing, you know, because open source and working in such decentralized research, working in a IP solution, because for them everything is, you know, a little bit, I think a lot of people in Global South can relate to that, you know, it's everything about making some form of money. And then they really agreed to that, because it seems for them, like for this decision maker, they clearly saw that this is a problem for us locally, and just be, you know, good and then solve our problem. We don't have to think beyond that. And as a country like that, they, even from the government side, they said, yeah, perfect, let's do it. And that's how they also help us to go to the clinical validation there. And again, another offer we always give to people is, since we have access to this BSL-3 lab and the clean room and the real life patients, if anyone wants to test or have access to something, we could probably help you. Thank you.
Yeah, and I can add from the German perspective. So we are also trying to work politics, but actually it's not that easy, because the gatherings of the commercial diagnostic labs are trying to inhibit our outreach in politics pretty roughly. So I think in one way it's a good sign that they see us somehow as competitors, but in the other way, it's it's sad to see, because we are not saying it's a diagnostic test, it's a screening effort for helping politics or helping nations to tackle the situation. And yeah, there's actually no competition, but still they try to inhibit us. It's, yeah.
Yeah, so I have a friend who's a dean at a local university in the city and in Chicago. And she's asking me, like, can we use your test already? Like, do you trust it enough? And I mean, I'm cautious, because we have not validated it on clinical samples. And, you know, that's definitely a hurdle. But we're getting ready to, you know, one way or another. But, you know, she said their health center at the university has a really hard time even buying tests to test people. You know, even the commercial ones, you know, that's definitely a problem, is even the access, or the access to the kits for extraction, you know. So there are so many problems. Like, I'm sure there are lots of kits, tests that have EUAs right now that can't actually be performed. Number one, you can't buy them because the big companies have bought them out, or the government has bought them out, or a component that you need is not available. So, I mean, there are a lot of things to work out, but we're doing the best we can, I mean, collectively, and I mean, it's great that we have JOGL, and it's great that we're all in this X Prize, because it's a really, I mean, Thomas, you're always saying this, maybe you said it and I missed it, but it's really an amazing thing for open science that we have 4 open source teams in this Ansari X Prize for rapid COVID testing right now. I mean, I don't know that there are any other open teams of the 200 and something that are, you know, in the competition at the moment.
I don't think so either. I'm going to pitch in here because what's been amazing is when you take a step back, what's going on with your project and these projects is, you know, if we contact them right to the end of being fully validated, documented, they become automatically something that people can build for themselves, trusting the system. And so, as you said, we're not, you know, you're not competing with companies because you don't want to, you know, producing those kits and distributing them everywhere. That's could do that. And so by providing them the opportunity to do it, that's fantastic, because I think we're going towards a model where, you know, having a collaboration between an open community that does open R&D and companies that, you know, respect that open aspect of the R&D, but, you know, manufacture and distribute the technology and pitch back to the community is going to be very very interesting. I don't know that many. Actually, I don't know much examples. Most of them are in the software industry and a bit of the hardware industry. But in the biotech industry, that doesn't actually exist. And if we really go right to the end with, let's say, at least those diagnosis tests, that will be a foundation for the existence of other diagnosis tests for other kind of you know, sickness, and this could have like dramatic effect overall. Like it's the first break, kind of what we see with iGEM. It could be exponential. So yeah, it's very exciting. Being able to get a PHP and the guys in Chile and in Sri Lanka to really be on our side and be excited about providing clinical trials and some kind of clinical validation has been really important. And they don't do it. If I talk about a PHP, they're not doing it because they say, okay, you know, we do. Why not? They're truly excited by these opportunities. They really see the value of what those projects are doing. In fact, they had the opportunity, other companies or laboratories, and had to refuse some of them because they had to choose basically. They couldn't work with everyone, and they preferred actually choosing the open version. So because they see the value being created and they prefer to be associated with that because they have also a public mission.
Thomas, your mic is mute.
You're muted, Thomas.
Oops. Yeah, I believe we have 5 more minutes before the end of this session. Do we have more questions for speakers?
I can add something to what you just said. I think we're getting to the point where we should be maybe thinking more about this, because even if we have a kit, how do we actually make it? How do we distribute it? How do we, you need all kinds of ISO, you need manufacturing. So we need to think how we can kind of take these biotech things and scale them up and really make them locally in ways that are sustainable somehow.
Yeah, I mean, the quality is always the biggest issue. Yeah. You can provide information on how to do something, but the results, if people are not following the instructions, where if something is a kit and it's all coming from a single source that can be held accountable for the quality, that's always the big thing with distributed biotech that I worry about. and maybe it's because I have been an FDA regulated industry and I know to what lengths that they go not to kill anyone. So, and freedom to experiment on your own and do stuff, I think is super important. But I also struggle with the idea of how do we make sure. I mean, one of the reasons why we're doing all this stuff across labs is we're trying to make sure that it works in everyone's hands, and that it's, just the way that, I don't know, if Carolina Biological made another kit for high schoolers, they would want to make it bomb-proof so that no matter, which teacher tried to do it, that it would actually work. Except the stakes are higher.
Luckily, also Guy's GMO detective, he's done a lot of work over the last couple years, getting this so easy that anybody could run the experiment. So that's sort of why we really got inspired by that for Corona Detective. But that's not the same as making it and producing it at scale. And that's where, we need nucleotides and we need primers. We need so many things to be right for it to work well. So,
and the quality control is really important. It really, really is. And yeah, we need to all be thinking about that.
Well, everyone's got to wish us luck in this X Prize thing. I mean, we have 4 chances that one of us is going to make it through. And the exciting thing about it is the next round, they will basically give you $50,000 to produce enough materials to send to people who are going to actually do clinical validation on your methodology. So that would be really huge if we could get at least one of the teams to that point.
And I mean, yeah, we will be still working together, all the teams on the goal, right?
And I see everything kind of coming together over particular circumstances, like the best test for an area that has a lot of technology, the best test for an area that doesn't have a lot of technology, the best test for places that can source things fairly cheaply and easily. The easiest test and the cheapest test and the test that might make the most sense in every country's rules and regulations, atmosphere, in whether or not it's a high complexity lab place, a low complexity lab, whether or not certain local resources are available. I don't think that there's going to be, one test to rule them all. So I think we will come together on certain things, but we may end up with a suite of different solutions, which would be really cool. You can imagine one for a country that maybe it was relatively easy to get the quasar reagents, and another for a place that might be able to synthesize oligonucleotides, but they might not be able to get the ones that are that have the the modified. So, or a place where, I mean, Guy's done a pretty good job of making a fluorimeter available to everyone. but you know what I mean. There might be different strokes for different folks, and I think it's great that we're all working on slightly different variations that might be the best in different, different areas and different circumstances.
So we'll have to wrap up. I would like to thank our speakers with the bottom of my heart. It's been not only a great session for me to hear also, but also it's just a celebration of the last 6 months we spent together. It's been really, really, really amazing. I think this challenge would not have been possible with you guys. I mean, you've brought not only energy to your own project, but the project of others to the community, to JOGL, and so it's I'm to to have you part of this global family, and I think we're really, as you've said it even better than me, and we're onto something here, and I think we're just providing the best, one of the best proof of concept that science can be done differently and efficiently, and so we need to scale this, and so. Yeah, wish us luck. Come on board, and let's change science forever. Let's make it not only open as it is, meant to be open today, but let's really make it truly open for anyone that wants to play a role in this great adventure. So thank you so much, everyone. And if you have any questions about the projects, you can actually contact them directly over to JOGL on the JOGL platform. You can, send us messages, and I'm looking forward to see you all there. Thank you so much.
Before you go, could I've seen in other sessions, like at the end that
we're
people
supposed to take a picture. Let's take a picture.
Take a picture.
Great idea.
You put the group up instead of just like.
Thomas loves to do this anyway, right?
Yeah.
So.
Yeah, that's my favorite game. After each of the, yeah.
I also would like it if people could unmute and if you want to give Thomas.
Oh, nice.
Yeah, if you can unmute, I believe you can unmute off. Yeah.
Thank you so much.
Thank you.
Thank you.